So today is the second part of my presentation at this conference. And as I mentioned last time, I started out yesterday talking predominantly about structural networks and model organisms in particular, and connectomics, which really has its origin in anatomy and structural connectivity. And today I will turn a page and talk about functional connectivity, which is perhaps more popular in some ways, more widely applied, certainly, I think, than structural connectivity in the field of neuroscience. And I want to begin by highlighting some contrasts between these two modalities, which I think are important to keep in mind. So a few contrasting features and properties between structure and function in the domain of connectivity. Using a nice overview diagram here from one of Carl's review articles some time ago, just to remind us, in the nervous system, we can measure and observe neural activity at different levels of scale, all the way from synaptic connectivity to connectivity between single neurons, circuits, and then of course the whole brain, which is the focus of my work and the talks today and yesterday. We can observe the brain with different modalities, from structural modalities that look at fiber pathways and tracts, such as diffusion tensor imaging or DTI, to modalities that observe brain activity through metabolic hemodynamic signals, such as the fMRI, functional magnetic resonance imaging, or EEG and MEG at the electrophysiological level. And these different observations give us different modes of connectivity, structural connectivity and functional connectivity, and effective connectivity, which I will leave aside a little bit today, in part because it's not maybe as prevalent in the field, and perhaps Carl will talk about it in his talks coming up later this week. So structural and functional connectivity, I've shown this slide before. I just want to show it again because it's so fundamental and highlights the contrast between these two domains. Structural connectivity on the left, proceeding through measurements of anatomy, either invasive histology, model organisms, or non-invasive imaging, as in humans, resulting in a sparse, typically sparse wiring diagram, connection matrix that assesses or reports the strength and sometimes the direction of structural pathways between brain regions or individual neurons. On the right is that whole big domain that I've left aside so far, but will focus on today. And that is time series recordings of neural activity, which can be carried out with EEG, MEG, fMRI, invasive electrophysiology, local field potential, single spike recordings, what have you, all of which result in time series, neuronal time series that can be recorded in the spontaneously active brain, or in a brain that is engaged in a specific task or cognitive function. Those time series can be analyzed with, a very large set of methodologies. Time series analysis is an old field. Everything as simple as a cross correlation to report similarities among time courses over time, or sophisticated things such as transfer entropy, I mentioned last time yesterday, and things like effective connectivity, which really are also based on observations of time series. The outcome of that in the domain of human neuroimaging very often is as simple as a correlation matrix, literally a Pearson correlation of time courses that are measured with, for example, non-invasive fMRI recordings. And this correlation matrix has fundamentally different properties than a sparse matrix derived from histology or anatomy. I want to stress that again, graph theory, very beautifully applied and applicable to a sparse matrix, weighted, directed, undirected, doesn't matter. There's different versions of graph theory we can deploy. to me, it's a much less clear match with the functional connectivity world, and there are things that we have to be aware of. So a few um, commonalities of the two domains of structural and functional connectivity are both can be summarized in a network representation, fundamentally a collection of nodes and edges between them. Both have topology, adjacency matrices, things we can look at with tools from network science, multi-scale typically. That means there's organization on multiple scales. We've talked about this yesterday. And community structure is also common to both. And I'll say a lot more about communities and modules in functional connectivity later today. But there are also many differences. Structural connectivity is fundamentally a different construct. It reports on, if you wish, on reality, on connections that physically exist, that have material basis. They are there or they aren't. And you can verify objectively, presumably if your methodology is good, or whether they exist or do not exist. There's a finite set of elements. It's a large number of elements in terms of synapses and the numbering in the trillions for the human brain, but it is finite, it's not infinite. There are complex attributes of density, strength, conduction speed, biophysics that are attached to each connection. They change across time. In the course of development or even lifespan in the humans, and of course in the course of plasticity. And they shape the flow of neuronal signaling. By and large, and there may be exceptions here and there, but by and large, neurons communicate through axonal signals that travel in the structural connectivity. That's the only way they have to influence each other. And so that is the skeleton on which neuronal signaling and communication must unfold. Functional connectivity is very different in the way that it manifests itself. It is fundamentally a statistical construct based on time series that we observe, recordings. It is dynamic, it changes from moment to moment. I'll show you later on how fast those changes unfold, even with a relatively slow methodology such as fMRI. Because it changes all the time on a time scale of below a second, in terms of hundreds of milliseconds, there's a very large and virtually infinite set of network configurations that functional connectivity can adopt. And of course, it also has complex attributes. Each edge in a functional network has strength associated with it, directionality sometimes if we have sophisticated measures, and it may persist or it may disappear. There are rapid changes across time, moment to moment, hundreds of milliseconds in EEG, MEG, seconds at the MR level, and connections, functional connections are highly input and task dependent in ways that structural connections are not. Functional connectivity, in my view, I'll come to the end, at the end of the day, I'll wrap this up and show you an overview of how I look at this. In my own view, functional connectivity reflects the outcome of communication events in the brain that travel through the infrastructure. So both of these domains are now commonly summarized under the rubric or headline of connectomics. That's just the way the field has developed. I still maintain to this day that the connectome as a finite set of, as an inventory of connections really applies most directly to structural connectivity. When we take into account these differences of structural and functional connectivity, that this has ramifications for how we should define simple concepts such as network hubs. This gets back to a point I made yesterday at the outset. We cannot blindly apply, or should not blindly apply, network science tools or graph theory to literally all the data that there is. We have to be aware where our data comes from. And in the case of structural connectivity, a few examples shown here. If we want to identify central parts of the network, nodes that are centrally positioned in the network in terms of its topology, that have many connections, that are between many of the other nodes in terms of shortest paths, we can do that. And in fact, if we do this in different studies with different imaging cohorts, different scanners, we find a pleasing level of convergence really across different studies with, for example, structural connections having high degree and high betweenness in multiple studies in the medial parietal cortex or the precuneus region, which is a region that is very highly connected in structural terms. When we go to functional connectivity and we record time series with fMRI, we really should not, in my opinion, in an unqualified way, apply degree and betweenness to those same data. We should actually look at this differently. And Jonathan Power, in a paper a long time ago now, pointed this out and came up with a way of defining hubs that is more consonant with the modality and the way that the data are actually acquired and processed. And that has to do with participation. What he suggested, and I think he's right, is that we should take a modular decomposition of functional connectivity. We should divide that functional network into modules or communities, and then look for nodes that have high participation, shown here at the bottom plot in warm colors as opposed to cold colors for those with low participation. Why should we do this? Because those nodes shown in orange and yellow and red down here have connections that range not only highly connected within each module, but also connected between modules. So they form bridges, functional bridges between different communities. And the methodology of participation coefficient, the way it's computed, the way we derive modules, this is something that is grounded well, I think, in functional connectivity data. It's a cross-correlation matrix, fundamentally a statistical assessment of similarity of time courses, and this approach, I think, works well. So we have to be careful. Our methodology, our analytics has to match the origin of our data, and that is one of those domain-specific aspects of network neuroscience. A few more words about hubs and their relationships and their association in both structural and functional connectivity, because hubs are a critical a component of the neuronal architecture and really a corollary to modular arrangements. You see on this diagram here on the left a very schematic little sketch of a modular network. And the nodes shown in black stand out because they're the only nodes in this network that actually have connections between modules. This is important for communication for obvious reasons between these different communities. Communities like this in the brain, human brain particularly, are often containing regions that are functionally related: visual systems, motor system, saliency, default mode, what have you. And it's therefore extremely important that these different communities, which are functionally specialized, are also integrated. So that's an arrangement that we might find. But here's one in the middle where we've added a few more connections to connect these hubs more directly with each other. And that takes this organization to a new level in some ways, because now these hubs can freely exchange information amongst each other. Each of them has access to their own community, and each of them also has access to the community of hubs, which combines highly processed, highly integrated information from different communities together. And that arrangement, I think it makes sense even just visually, seems ideally suited for integrating information in a network such as the brain. And that's where I started out together with Martin Mannenheubel to look at Rich Club organization in the human brain. We found it on our first try. Highly connected brain regions shown here in red in this human brain network. Down below, they are isolated so you can see them a little better. They comprise regions such as the cingulate cortex, the medial parietal cortex, the insular and the medial temporal lobe in both hemispheres. As you can see down below in this plot here, they are widely distributed across the human brain. Remember that plot I showed you yesterday of the macaque brain, similar arrangement. Hubs are widely distributed. They are distant from each other in terms of metric distance. And yet they are highly connected. The density of connections between these nodes exceeds that density that we would expect based on their degree alone. And that is illustrated in this diagram up above, which shows us the excess of connections beyond chance, beyond that null model of degree preserving random network that we find in the actual brain. So yes, the human connectome exhibits rich club organization. As I illustrated yesterday, rich club organization has also been found subsequently in other organisms, such as the nematodes, the elegans, Drosophila, the rodent brain, and so forth. Interestingly, this was the first demonstration of it in any nervous system that you're looking at here, and it's actually held up well, given that other methodologies of acquiring anatomy are often considered to be more valid and more accurate. Couple of predictions that come from this arrangement. If we want to travel from any node to any other node in the human brain network, a surprisingly, perhaps surprisingly high proportion of communication paths need to travel through that rich club network subgraph. 2/3 have to travel through rich club edge. Almost all of them have to access the rich club somewhere, and that's an analogy to the transportation network, really, where the highway system that we have, really every country has. We have it in the US, of course. You have it in Russia as well. Serves to connect widely dispersed big cities, and is often used for travel between these cities, and actually absorbs a high proportion of vehicle traffic. So we sometimes refer to the rich club as the highway system of the brain for that reason. Another prediction, and that's actually borne out to be the case in subsequent follow-up studies, is that if we damage the rich club, if we delete a node or multiple nodes in the rich club, or connections in the rich club are damaged or diminished in strength, that should have a disproportionately large effect on network integrity and efficiency, the ability of the network to communicate. Because these nodes and edges are so central, so important for communication, if we damage them, this has a disproportionate effect. That's the prediction, and it's turned out to be quite true. We have found this with Martin Mannenheuvel in data sets to do with schizophrenia, and others have reproduced this. The importance of hub regions in general as points of high communication, but also as points of high vulnerability, was shown impressively by Nick Crossley in a meta-analysis a few years ago. He looked at anatomical scans of individuals who suffered from a range of disorders. You see the disorders listed here on the left. There's almost nothing on this list that we're not familiar with: autism, schizophrenia, Alzheimer's, obsessive compulsive disorder, post-traumatic stress. All sorts of brain disorders are captured by this meta-analysis. In fact, over 20,000 individuals' data has been entered into the analysis. And there is a distinct skew towards the high degree side, the hub side of the connectivity in this analysis here, which is an analysis that asks the question, where in the brain do these conditions start? Where do we see the first signs of atrophy and physical damage to the architecture in anatomical MR scans? And in most disorders, not all, but in most of them, the disease appears to involve appears to disproportionately involve regions of the human brain that have high connectivity. So that's an interesting, opens an interesting perspective. What's cause and what's effect is the high degree itself a vulnerability that predisposes certain brain regions to be more susceptible to damage, or is the fact that is the very fact that high degree nodes are damaged, does that give rise to a disorder? Which is cause, which is effect? They probably go together to some extent. To summarize this slide, across the clinical literature, there's been a lot of focus on looking at network hubs and high centers of high connectivity in the human brain to look for potential pathophysiological effects and damage-related effects to damage-related effects on cognition and behavior. Now, leaving aside, by the way, this clinical area, let me go back here one moment. The clinical applications of connectomics, especially human connectomics, are really an area all onto the, it's a very large area now. I have worked and continue to work with people on many of these disorders that you see listed here on the left, currently with an emphasis on Alzheimer's disease and dementia. And we have learned a lot about the relationship of connectivity and networks to clinical manifestations, clinical disorders, and it's a huge area. I'm sure in your country as well, here in the US, very much on people's minds because of the prevalence of many of these disorders and their societal and economic and health impact. So we continue to work in this area with our colleagues in clinical institutions. Now turning over a new leaf, and this is sort of the main part of today, turning to models of brain dynamics. I showed you this slide already at the end of the talk yesterday. I want to briefly reiterate with you what motivates this. On the left is our structural network, and that's what I spent all of yesterday, telling you about. And in first approximation, this is not entirely correct, but we may think of it as fairly static or stable across time, at least certainly on the time scale of minutes to hours to days. At the level of these axonal tracts, there aren't many changes that occur on those relatively fast time scales. But as I illustrated earlier today already, functional connectivity, the ebb and flow of activity itself, and the relationship, the bivariate, statistical relations among time courses, those change all the time. They change when the brain is spontaneously active, as we see in this little movie over here. And they certainly change when the brain is engaging in task-related activity in behavior and cognition. So how do we get from one side to the other? That's the question mark here. And that's been occupying me and my lab here for the last, I would say 20 years. It's one of the key questions, I think, in neuroscience. How does structure relate to function or dynamics or behavioral outcome? How do we get from neurons to behavior to cognition? That's ultimately the big challenge for all neuroscientists, really. And this question here, in a more crystallized context in terms of human imaging, how do we get from anatomical connectivity of tracts and pathways to something as dynamic and flexible as functional activations and their relationships? That's where we've been applying a lot of our time and effort in the last a couple of decades. What is functional connectivity and how do we compute it? We take a movie like what we've got on the right. That's a recording of an MR in an MR scanner of a person undergoing rest, what we call rest, which is spontaneous brain activity. We can discuss later what rest really means. And we take frames of this movie, essentially snapshots in time of what the activation state was, and then do something as simple as a cross correlation. When you do that, and it literally is as simple, embarrassingly as simple as a Pearson correlation of the bold time courses, D-scored, we get a fairly consistent picture across individuals and across imaging centers and participant cohorts. And that's surprising because when you think where this comes from, so-called resting state is a totally unconstrained, situation. You literally put a person in a scanner and you say, please don't fall asleep, keep your eyes open or closed. In some cases, they're closed. And actually, some people say, think of nothing in particular. And that's it. And we record for 10, 15, 20 minutes, sometimes longer, sometimes less. And we take those time courses coming from the brain, cross-correlate them, and then remarkably, a consistent picture emerges. Even though what every person was doing in the scanner is unconstrained, and is not controlled or subject to instruction or manipulation, over a long period of time of minutes, of several minutes, ideally in half an hour or an hour even of recording, we're seeing very consistent pictures that emerge, and they look like this. Here we have a cross correlation matrix where high correlations are shown in hot colors and low, strong but negative correlations are shown in cold colors. And the blockiness along the main diagonal is no accident, because we've actually grouped together these nodes, which are brain regions in the cortex. We've grouped them together by a generally accepted convention in our field of how we name different coherently active subsets of nodes during resting state. Control network, default mode, dorsal attention, limbic system, ventral attention, somatomotor, and visual networks are captured in this diagram. This nomenclature comes to us from large, very large studies, and carried out in some cases with thousands of individuals, that take resting state and construct group averages that capture the consistent pattern across individuals. These two studies here, shown side by side, came out almost simultaneously, taken in different imaging centers with different participants and different imaging setups, different magnets, different pre-processing and what have you. And yet they produce a very consistent, I think, picture. As you can see, I invite you to just compare the salmon colored network on the left, which is the default mode network, to the red colored network on the right, which is also the default mode network. And you see that, you know, despite these differences in imaging centers, participant cohort and whatnot, these are very consistently captured. Now, these are group averages over many individuals. A lot of the interest in human functional connectivity analysis goes into studying individual differences. This is a much more challenging problem in some ways, because we're now caring about how your brain network differs from mine. And no brain is identical to any other brain. Brains are highly individual. Even in monozygotic twins, brain structure, even the level of gyrification, the folds that you see on the surface looks quite different. And so we care a lot about individual differences in our field. They are unfortunately very hard to reliably ascertain, because you would need very long-lasting, long-duration recordings from each individual to really establish them well, which are often not available. So I will leave that aside a little bit for the current today's discussion. That will be a third talk about individual differences. But it is something that motivates a lot of the studies in functional connectivity and human neuroscience to establish differences between individuals. Now, where does this pattern of functional connectivity come from? And we had the intuition long time ago now that the resting state is a reflection to some extent of the underlying anatomy. How did we come to that hypothesis? We observed a strong, robust, but not entirely simple relationship between structural connectivity and resting state functional connectivity in studies that compare the two in animals, in the macaque monkey shown below, and also in humans in our own work with Patrick Hagman and Chris Honey. And we also know that resting state networks such as default mode, control, visual, etc., that I've shown in the previous diagram as coherently active and coherently coactive sets of brain regions, that they're internally linked with structural projections. If we cut structural connectivity, in this case down below, in a single individual that underwent surgery for epilepsy, and where the corpus callosum was severed, the pathway that links the left and right hemispheres to each other, there was an immediate change in functional connectivity following that manipulation. All of these lines of evidence point to a role of structural connectivity in shaping and molding functional connectivity. We also know this from modeling. Here, the work that I did back in the day with Chris Honey, my graduate student at the time, structural connections of the type that I've shown you yesterday. This is macaque monkey cortical connectivity, can provide a coupling matrix for a set of biophysical equations that at each node report or capture how that node behaves through time. In this case, the equations are quite detailed, have actually individual ionic currents in them, and voltage sensitive channels and so forth coming from electrophysiology. These biophysical equations operate at each node, and they are coupled by this coupling matrix up above. So essentially, that sets up a system of coupled ordinary differential equations. So now we can integrate those ordinary differential equations, and we can pass our simulated time series through a synthetic bold mechanism to create synthetic bold data, which we can then cross-correlate using the same post-processing strategies as are done in empirical data. So effectively, we are doing a simulation of a brain without using a scanner at all, and create synthetic bold signals from that brain simulation. It turns out that those approaches are quite useful because we can perturb this matrix, right? And once we have a simulated a brain simulation, we can manipulate that. We can perform lesions. We can delete or even add connections to that matrix and see what the outcome is in terms of dynamics. And we can even put it through a task-related paradigm by providing inputs to individual nodes that simulate task effects. Remarkably, even though this is an extremely simple approach, coupled ordinary differential equations and a synthetic bold mechanism on top, the relation to empirical data is actually quite strong. That's without any data fitting. That's without any explicit, machine learning was not a thing back in the day when we actually performed these studies. It didn't really exist yet as a scientific paradigm in our field. It does now, but not back then. So we did perform no data fitting at all. What can we do with models of this type? Let me just go back one moment. This type of model we have currently, we have actually returned to this year. And I'll show you at the end what we can now do with this that we couldn't do back then, or that we didn't capture back in the old work. There's some interesting new wrinkles that I will come to in just about 10 or 15 minutes. In the meantime, though, there's other modeling approaches we can pursue, more abstract. I was interested at the time when we did this work, not so much in making these models more realistic. Other people have done that, creating more detailed models, larger models, models that have individual neurons in them, models that have more parameters. I wasn't so much interested in that, to be honest with you, because for me, a model ought to be simple. It ought to be capturing something fundamental about how the system is operating, but it should also do away with a lot of the excess detail that we don't really need to explain a phenomenon that we're interested in. So, to paraphrase an old saying, a model, in my view, should be as simple as it can be, but perhaps no simpler. A famous person once said that. So here's a different modeling approach that we pursued coming out of this work, and that has to do with communication, a topic that continues to fascinate us. The brain is a communication network, right? What neurons do, in some ways, do best, and what sets them apart from other cells in our body is that they communicate with each other along a very specific set of connections. No other tissue does that. The liver doesn't do that. The skin doesn't, the stomach does not, our musculoskeletal system does not. Even though all of these cells have their own excitable properties, brains communicate. And to me, that is the essence of what the brain is doing. So we wanted to build communication models. One way to do that is to ask, can we conceptualize information flow or signal spread in a network shaped as the brain? And one way that we can do this is we can take a path going from a source to a target, and we can say a signal travels obviously from source to target along a set of connections. And if that set of connections is stronger, here it is weaker in the second example, the functional connectivity we observe, the similarity of the time courses that we may record from source and target, will be stronger in case 1 over case 2. That's what's illustrated here. Functional connectivity for this arrangement ought to be stronger than this one. If the path gets longer, if there are more intermediate steps, once again, communication should gradually diminish, right? The faithfulness with which a signal is transmitted from source to target should decline with the number of intermediate steps. A final step here, if we have incoming and outgoing connections on intermediate nodes, we would expect prediction that the more divergence and convergence exists on the intermediate nodes, the more ways there are for a signal to go astray, to diffuse outward and away from the path that it's intended to travel, the functional connectivity should diminish. And these aspects of connectivity and communication, my former postdoc, Joaquin Goni, now professor at Purdue, we set out to test whether those structurally determined characteristics of paths are actually predictive of functional connectivity in empirical data. So we built an analytic model, a model that runs in MATLAB in under a second based on graph theoretic concepts such as path, connection weight, path length, search information, and path transitivity, and looked whether or not it would predict functional connectivity quite well. And it does, and it did. This is a somewhat fanciful plot where we show on the left side a functional network that was empirically recorded. On the right side is the prediction of that functional network based on its underlying structural anatomy. As you can see, that relationship, again, without any explicit tuning of parameters, just a multilinear model with 4 beta values, we get a very high agreement between these two. So the model, analytic model based on graph theoretic communication measures can predict functional connectivity very well. Over here on the right are some examples of particular brain regions. Other work carried out by my then postdoc, Vladislav Misic, now professor in Montreal, looked at did a very different approach to communication, more like as a message passing and buffering and switching network, which he implemented using an ingenious communication paradigm that was coming in some ways from discrete message passing networks such as the internet. And if we look at the predictions coming from a communication model such as this one implemented on the structured connectivity, there are connotations between there are associations with brain hubs that we know exist in structural connectivity and dynamic hubs which attract a significant proportion of signal traffic. When that happens, the signal traffic that gets attracted at these hubs can actually result in bottlenecks where communication can slow because of overburdening of these nodes with incoming and outgoing signals. Finally, a very simple communication model based on diffusion. If we take 2 nodes at random in a connectome network and we inject different colors, think of them as diffusing colors coming from those particular seed points, red and blue in this case, no connotation here with politics. We've got red and blue spreading outward, and in the connectome, other nodes will now convert to those colors as they are affected by this outward spread. And the question we ask is, where do these 2 signal fronts or cascades, where do they collide? What are the points of convergence for these 2 signaling cascades? The heat map for that, shown down below, correlates very strongly with multimodal higher order association networks such as default mode and dorsal attention that are known to be critical for more integrative cognitive processes, while unimodal, smarter motor, ventral attention visual systems are less well captured. So the framework here is one of communication dynamics. I look at the brain in the following way. Structured connectivity, the connectome is the basis, the foundation upon which our field really rests, and networks, of course, loom large in this context. I've talked about this all day yesterday. On top of that, we have an ebb and flow of signaling events that occur along axons that will travel from certain sources to other points in the network through pathways. These pathways are structural pathways that define axonal multisynaptic paths. And the result of those signaling events are statistical alignments that we can record using fMRI, EEG, MEG, neural recordings, what have you, of time courses, functional connectivity, a simple correlate of that. So we have good data, I think, in anatomy. We have good data in functional connectivity. We don't really have a lot in the middle. And I think this is probably the domain of effective connectivity that's still very much underappreciated. What are the causal pathways along which neurons and brain regions signal to each other? We don't have a very good way of directly measuring those. We have to infer them quite often using time series data and anatomy in the conjunction of the two. But fundamentally, that's the picture that I still have in my own mind that I like to sort of use as a framework for how to approach networks in structure and function in the brain. The whole paradigm of communication dynamics is evolving. Our article back here a few years ago now kind of set the stage for that, but it's evolving quite rapidly in the past few years. And the jury as to which communication protocol is implemented in the brain is still out. We don't know this yet. But just to illustrate to you what some of the alternatives might be, I talked a lot about shortest paths also yesterday, and in historical terms, they've been a real focus in our field. But sending a message along a shortest path in a complex network implies that you have full knowledge of the connection topology. Just picture yourself, you come to a new city, you arrive at the airport, you're looking for a way to get to your hotel, you look at a subway map, and you try to figure out what's the best train to take from the airport that gets me to my hotel, maybe with one or two connections in between. You look at the map and you can actually determine that, but the brain cannot look at a map and determine how to send an impulse or a signal from point A to point B along our shortest path. That's not possible, right? There's no known mechanism by which neurons or brain regions have access to that global knowledge. So while routing is appealing in many contexts when we try to navigate networks, communication networks, transportation networks, etc., it is really not an option for the brain because the brain cannot access its own topology. Instead, an alternative would be diffusion. So the brain just sends out, each brain region sends out signals along all its outputs, period. It does not care or does not know where those signals might go. That's diffusion or random walk. A random walk model. The information cost is minimal. In fact, the brain does not need to know anything about its own topology to do this. But of course, it's very wasteful because signals are dispersed in all directions and can therefore also scatter and interfere with each other. And on this diagram down below, in terms of information cost, information meaning the amount of global information about the topology needed, routing is the most expensive, diffusion is the cheapest. But diffusion is most wasteful, while routing is, of course, the most efficient in terms of minimizing resources for communication. So where's the brain on this spectrum? We don't know, honestly. It's an ongoing research effort also in our lab and with collaborators overseas. Does the brain use routing, or is it possible for it to use routing or routing-like strategies to communicate? Does it use diffusion in other contexts? An open question, but one that we can now finally begin to answer given the modeling approaches which take an important role in this effort. So now I'm coming to the last part of this and talk about work that we've just recently started here in the lab, working with now my colleague Rick Betzel, my grad student Josh Faskowitz, you'll see in a moment. And that relates to dynamic functional connectivity. Already in the work with Chris Honey, we noticed that when we look at simulated brain activity, here a simulation of a macaque cortex, and we take windows out of this activity and we look at how the topology of functional networks changes in the sort of windowing fashion, it changes quite a bit. So if we take a small snippet of activity and we compute functional connectivity on that, and we slide that window across time, the attributes of nodes and connections over time will dramatically fluctuate. Here, for instance, the centrality or hubness of areas such as visual area V4 or prefrontal area 46. They fluctuate from being strong hubs in case of area 46, you see some peaks here, to being almost non-evident as hubs in the network at all. And this is changing on a time scale of multiple tens of seconds, a very slow time scale, actually, interestingly. We called that at the time hub dynamics, the changing status of a brain region in the network over time, dynamics of hubs. And there's been a long line of computational, theoretical, empirical studies that suggest that indeed dynamic functional activity exists in the human brain and in other species, and that in fact we can characterize these dynamics as being multi-stable, near critical, and so forth. Here's a picture, a movie of a windowed functional connectivity recording in a human brain. On the left is the grand average of all time points, the functional connectivity cost correlation network observed over many minutes. Down below is a movie of a frame-by-frame account of what goes on, and that is captured at a time scale of a few seconds. Work by Alex Jones 10 years ago now. We had done similar things in our own lab, but never published them, unfortunately. So that network down below, essentially think of it as if you take a superposition of those pictures you saw in that movie, you would be getting the picture on the upper left. But at any given moment in time, the network undergoes very dramatic shifts in terms of its network structure and topology. And there have been many studies since then. I don't even want to go through this list in detail. Yes, sliding window, cost correlations show significant fluctuations over time. There are network states, as they have been called, which are repetitive patterns that occur in such a movie that we can cluster and identify as belonging to a particular class of configuration. There are differences in dynamics that occur in psychiatric disorders, development, consciousness, and during ongoing cognition as well. A huge literature that has ballooned over the last several years. I stopped adding references here a few years ago because it became an impossible task to keep up with it. Suffice it to say, abundant evidence that dynamic functional connectivity does occur, and we can measure it and analyze it with a number of different tools. I'm going to go forward here to work that we've done in our lab with my former postdoc Makoto Fukushima, and we focused on the following: modularity, right? I mean, remember I talked about this a lot yesterday as a fundamental property of how brain networks are organized, and it also happens to be one thing that we can measure appropriately and track in functional connectivity over time. So Makoto set out to look at modularity in windowed functional connectivity recordings to see how it fluctuated across time and what we could learn from those fluctuations. Applying a null model where accidental fluctuations are excluded from analysis, he focused on extreme excursions of high and low modularity and constructed summary pictures of functional connectivity during high modularity and low modularity intervals. And he did this across two different subject cohorts, large cohorts of subjects from the Nathan Klein Institute, NKI, and the Human Connectome Project, HCP. In our field nowadays, it is imperative really to carry out studies in large subject cohorts. I can't stress that enough because of the variability of human brains and the individuality of human brains really. It is essential to establish consistent patterns across large subject cohorts. Furthermore, different scanners, different processing strategies can result in different outcomes. And so it's important to establish that findings are robust with respect to variation in acquisition and pre-processing. This is something that plagues us really in the MRI, human neuroscience arena. We spend a lot of time in the lab wrestling with issues of pre-processing and acquisition and exclusion of artifacts and physiological nuisance variables. But unfortunately, in our field, that is par for the course, as we say. That is something we have to do. So going back to my Makoto study, 2 different subject cohorts, 2 different scanners, imaging centers, totally different. And when you look at the high modularity and low modularity states, Q standing in for modularity, our Q metric that I illustrated yesterday, we see 2 things. First of all, unsurprisingly, the high Q states are more modular. It's got to be that. It's got to be true. But more importantly, when you look in detail as to what systems are co-varying across these 2 different subject cohorts, there's a consistent pattern to it. But what Makoto found is that in high modularity states, the task negative systems such as default mode and attention systems, default mode and control systems, which are more highly engaged when there's no overt task demand, are highly covariant, and the task positive systems are also, but they are mutually disconnected. You see these blue splotches here, which mean anti-correlation. So visual, somatomotor, attention systems are decoupled from default mode and control. That's the signature. That's that inherent dichotomy that we see not just in the NKI set, but also in the HCP data set. Now that gets me to the present day really, and work that we've been excited about a couple of years now, working with my colleague Vic Betzel and Josh Vaskowitz, my grad student. We developed, I think, a fairly new approach to looking at fMRI dynamics. So we call that classic functional MRI, functional connectivity, is based on the similarity of activation patterns or time courses, and where similarity here is as boneheadedly simple as a Pearson correlation of the normalized or z-score time series. Despite its simplicity, this methodology has yielded, consistent results across studies and has been retained in our field. I should say that technically speaking, these are partial correlations because a lot of nuisance variables, age, sex, motion variables essentially have been accounted for and have been partialed out. What we did is the following. In classic analysis, we take bold signals, measurements of activation in brain regions at each moment in time, shown here for 2 regions. And we cross-correlate them, which is essentially we multiply element-wise each of the activations and then take the mean of that. What you see here is the step that has not previously been considered, and that is the so-called co-fluctuation amplitude. It is the element-wise product by element, I now mean time moment by moment product of the 2 normalized time series that you see on the left. The mean of this, what you see plotted here, the mean of that is exactly equal to the functional connectivity or Pearson correlation. One definition of the Pearson correlation is that it is the element-wise product of the normalized scores and the average of that, okay? So what we're doing here is we add nothing new to the Pearson correlation. We just take it back 1 step. We call that unwrapping. So rather than reporting a single value for the Pearson correlation between these 2 nodes, we report an edge co-fluctuation amplitude. This is an edge time series because it is a time series that unfolds on each edge ij separately. What this buys us is it now gives us an edge by time matrix. Okay, you see this matrix up here. This is essentially the Pearson correlation of functional connectivity unwrapped in time. If we take the mean along the edges and we wrap it back into square form, we get exactly the Pearson correlation, the functional connectivity that I've shown you all day today. Okay, or alternatively, if we take the Pearson correlation and we vectorize it and then unfold it in time, we get the picture on the left, or on the right rather, okay? Now, what we can then do, and in fact, Josh has done this in a nice paper came out in Nature Neuroscience last year, is we can take these edge time series and in turn create a functional connectivity from them. We call that the edge functional connectivity, which is now a similarity of the edges, not the nodes. This gives us a much larger matrix, which is a little bit of a nuisance with respect to analytics, but that's the way it is. And we can then do interesting things with EFC, look at communities which now have overlap on nodes, reproducible across different cohorts, and show interesting patterns that relate to differences in some other sensory attention and default control networks. And the papers that Josh has, especially the first paper in Nature Neuroscience, quite substantial, 20 supplementary figures, lays that all out in much detail. I will leave that aside and instead focus on the following for the last few minutes of today. You've probably noticed when you looked at this matrix shown here again, that there are these vertical streaks running through it, right? That's quite conspicuous. And if you are a seasoned MRI researcher, you get very paranoid about features like that in your data because they are often indicative of what we call nuisance variables of physiological noise, heartbeat, respiration, head motion in the scanner, things that we do not want to see in our neural recordings because we don't care about what the heart is doing or the breathing of the individual or its involuntary motion of the head. We want to know what the brain is doing, the neurons are doing underneath. So those are often excluded. When we saw this pattern for the first time, I remember this very vividly, we were all like very concerned because this could be motion corruption or heartbeat or anything else. It turns out it is not. We very strongly looked at this with recording of all sorts of nuisance variables and parallel when these moments of high co-fluctuation occur, when there is strong signals in the brain that co-vary along the edges, we call events. Nothing happens that we can point to that suggests it's an artifact. Okay, so these are not artifactual. In fact, anybody with access to MRI data from their own imaging center or public data can find these features with just a few minutes worth of MATLAB coding. It's super simple to see. Just regardless of that, no one that we know of has seen these before. So we kind of hit upon a phenomenon here that was very conspicuous and yet had not been seen before, which is sort of, it doesn't happen very often in your scientific career, quite frankly. The way we analyze these patterns is that we take the global magnitude of co-fluctuations, which is typically something we call the root mean square of the, or the root sum square in some cases, of the edge co-fluctuations at each moment in time. That defines these sharp peaks, which we call events. So I'll be using the term event from here on out for the rest of today, and that refers to these high co-fluctuation moments in time. First of all, just basic discovery, edge time series recorded with fMRI, we exhibit Barsky behavior or events in all individuals. They're unrelated to cardiac or respiratory cycles or head motion. And like I mentioned, we really see them in all individuals. This is not something that is expressed just in a few people. This is something that is universal. I might add that we also see them in animal data. Others have seen them in animal data, and we see them even in non-fMRI or non-MRI recordings. So this is not something specific to MRI or to humans. Furthermore, as I'll show you in just a minute, we also see these in models. Okay, so stay tuned for that. What does classic functional connectivity look like when events occur? So we take, to establish that, we take these snapshots of brain activity when events occur, and we average them together to see what the pattern looks like, effectively doing a decomposition of the full FC into components. And it turns out that the event frames really drive what we see in the full FC. It has a mathematical reason for this. It's not surprising in some ways. It wasn't something we had understood when we got into this, but of course, in hindsight, it should have been obvious to anybody for years, but it hasn't. Non-event frames contribute much less to the overall functional connectivity. And there's a common signature of events across the brain, which involves a decoupling of the control and default mode systems from those task-positive areas in the brain that we encountered before in Makoto's work. Remember that, with the windowed analysis, which is related. Okay, so now let me show you a movie. Here's a movie I've taken off a typical human connectome project subject. And we're looking at that person's co-fluctuation pattern over time. What you see up on top is the co-fluctuation matrix, which is positive in red, and blue means anti or negative co-fluctuation. And I've illustrated here the classic systems: visual, motor motor, dorsal attention, mental attention, limbic, control and default mode that we know from classic functional connectivity. Here's our edge time series matrix that I showed you a few times already, and you see these characteristic streaks that occur when there is an event. These events manifest in our movie as high red-blue, high contrast patterns. The point that I want to make with this movie is that if we take the images at the top and we just take literally just the mean, the average of them, we get functional connectivity. We get the construct that people have been working on for 10 years now, with thousands of papers that have been written about it. Thousands. I mean, I'm not kidding. Here we have unfolded it, and this is something we've never seen before, and the field has never seen before, and that's the moment-by-moment contribution to functional connectivity on each moment in time. Right? By moment, I mean in this case single TR resolution, single resolution of an image in MR, which is about 780 milliseconds in this case. It's a very fine scale resolution of dynamics. So we've added nothing to the bold time series or to the analysis really, except unwrapped it in time. And this is what you observe in a totally normal, totally characteristic representative recording of the human brain. We were drawn, of course, by these to these high co-fluctuation moments in time. What's causing that? What's driving this, and what is happening here? And the answer to that will be given by a computational model that I'm going to come to. I'm going to skip these, so I'm running out of time. So that's my last 5 minutes, and this is work that is submitted, not yet published, and work that I think sheds light on where these fluctuations that we see in dynamics come from. Since we are computational people in the lab, we wanted to look at our computational models and see whether we find events in those models as well. Why is this important? Well, if you can model something and you see similar things happening in your model to reality, no, it's not a proof that the model is accurate, of course, but it allows you to essentially come up with a mechanistic count of what is happening, at least in your model, what is causing the events in your model. You can't prove that that's the same chain of events that occur in the brain, but at least there's an existence proof, as you, if you wish. And we addressed this question by implementing two different models for functional connectivity. One is a phase delay Kuramoto-Sakaguchi model. For those of you who are familiar with this literature, this is a classic coupled oscillator model, single variable phase oscillators. Kuramoto has been studied forever. Very simple, deceptively simple model of coupled oscillators that is capable of remarkably rich phenomenology. In this case, a phase delay model where we incorporate the equation down below, a conduction delay that imposes a phase shift in the coupling term, by which we can handily incorporate the delay structure of the neural network in our simulation. We also implemented a second, more classic neural mass model, actually in the exact same model we used 15 years ago in the work with Chris Honey. Both of these models use the same 200-node SC matrix And we are then simulating functional connectivity and are looking for whether or not we find these short time fluctuations in the time series. Here's the essential sort of workflow, our coupling matrix up above, which has both weights and delays, chromotor oscillators, which are single phase that's rotating, time series passed through a hemodynamic response function to get simulated bold, from which we then compute edge time series, very much the same as we've done with empirical data. And we then get a blow-by-blow account of the amplitude of these edge time series across the whole brain. And you can see already in this diagram, there's an actual data set up here, that we do see peaks in the temporal dynamics. We adjudicate high peaks by using a time shuffled null model that we subtract and then retain those peaks that exceed the expected envelope based on the null model. So here in this case, we have about 10 or 12 events that have been identified in time. Summary of the model, structural connectivity weights and lengths shown up below, up above, and the empirical functional connectivity illustrated here. If we look at our simulated FC, it adheres quite closely to the underlying structural connection modules. This is our co-classification structural connectivity matrix, illustrating effectively where the modules are in the structural connectivity. Here's our functional connectivity on top. This is simulated now, and it matches quite well with the modular structure of the structural connectivity itself. The match with empirical data is, within ballpark of what these models are capable of, given that they are so simple. Here's the fundamental phenomenon that we had established right from the beginning. It's super simple to see in simulations down below, an edge time series matrix, exactly of the kind that I've shown you a couple of times now for empirical data, and down below the RSS summary with the events illustrated. You can see them running through that matrix in vertical streaks. Now, importantly, because this is a simulation, there's no scanner involved, there's no person, nobody's beating heart or moving head gets in the way, we can confidently exclude that those physiological variables play a role, right? Because there is no physiology. And yet we do see the same phenomena that we see in empirical recordings. We do see events, they occur over a very broad range of the parameter that gets varied, which is the global coupling constant. And it turns out they have very similar characteristics to empirical data as well. They approximate the full FC very nicely. They are more stereotypic with respect to each other. So events tend to capture patterns that are more similar across time. And they go together with low dimensional dynamics. That is, there is a high amount of variance explained by the principal component that adheres to the event structure itself. The final point is that if we look at the structural connectivity matrix underneath that is driving our activity, events that we can cluster together and define cluster centroids that way align very strongly, very significantly with the underlying structural connectivity. To put it differently, structural connectivity modules, modular structure in the anatomy, appears to be associated with, and we presumably can assume is driving, the co-fluctuation patterns we see during these short-lasting events. If we eliminate modules from the structural connectivity matrix by rewiring it, maintaining the weight structure and the length structure overall, events are basically abolished. Final point, if we wire a synthetic matrix, this is not a real brain, but a matrix that we can define using simple essentially a simple MATLAB function that defines modules ahead of time using 4 modules in this case, and we run that as if it was a brain. We get event clusters and we look at their alignment with the underlying modules. That alignment is very, very strong. So converging evidence here that the events that we've seen in fine scale dynamics of functional connectivity at the time scale of seconds are indeed driven by modules in the structural connectivity matrix. So it's the modular architecture of the structural connectivity that's driving the shape of these event patterns. And we, at least in our model, they are independent of any physiological or scanner artifacts. Because of the enormous amount of attention that's been given to functional connectivity in the field, thousands of patterns, I mentioned this. This new way of analyzing functional connectivity, I'm going to predict, will have a large impact because it now allows us to go back to already published work, to existing data, to look for these events, to see how they shape the underlying, the overarching functional connectivity, how individual differences play out during event timeframes versus non-event timeframes, and so forth. There's an enormous amount of work to be done. We are up to our ears on follow-up studies on this already in our lab and in Vic's lab, and are very excited about this new direction. Because it allows us, this is my final point, and then I'm done for the day, it allows us now to tap into, remember this diagram I showed it to you before yesterday, it's a diagram I like to come back to because it sort of reflects my thinking about modular architecture that I've been engaging in for at least a decade now. These fluctuations between states of the brain that are more, segregated, more modular in terms of modules being disconnected, versus more integrated, perhaps through a rich club architecture. That dialogue between these two, almost a dialectic between these two extremes, plays out in time in the resting brain, and we also believe in the task engaged brain. And I have a feeling it is fundamental to our understanding of how information is processed, how information flows, is shared, is integrated. So we now have a new tool, which is the methodology I just illustrated, to get at this using the most prevalent imaging technique on the planet, which is MRI. So very excited about the future prospect for this work. Finally, networks rule. They really exist on all levels of the scale that you see here in the brain, from molecules all the way up to the whole brain and the architecture of brain systems that I've been talking about a lot these couple of days. And it has led me to not only propose a new term, network neuroscience, for the field, but also found a journal that is now in its 5th year of publication. And we just got a very nice impact factor for it that I would welcome anybody in the audience who has related work to submit on the intersection between networks and brain, one of the most promising areas, I think, in all of neuroscience. So that's the end for today. Thank you very much. And I look forward to your questions. Thank you.